Hello, this is a video on action potentials in neurons and skeletal muscle, and excitation contraction coupling. These are the learning objectives that this video is based on, most of which are from Lecture 13, but there will be a bit of a preview on Lecture 15 since I will be talking about the neuron first. Let's go over the basic structure of a neuron. These branch-like structures you see here are called dendrites, and they are the part of the neuron that receives signals through various receptors. The cell body is the part that contains the nucleus and organelles. This long area here is the axon, which transmits the electrical signal that is received from the dendrites. Finally, these bulb-like structures at the end of the neuron are the axon terminals, which store and release neurotransmitters. If we zoom in on an axon, we would see that there are clusters of voltage-gated sodium channels, which have a structure that is sometimes informally referred to as the ball and chain. We'll see what this does in a moment. There are also voltage-gated potassium channels. These blue dots represent sodium ions, which have a higher concentration outside the cell than inside. The pink dots are potassium ions, which are the opposite and have a higher concentration inside the cell. What we can do with the neuron is measure its membrane potential, which is really just a fancy way of saying the charge inside the neuron. At rest, all cells, including neurons, have a negative membrane potential. However, if neurons receive a stimulatory signal at the dendrites, this would cause the charge to become a bit less negative. If this change reaches a threshold level of charge, represented by the dotted blue line here, voltage-gated sodium channels will begin to open. This causes sodium ions to enter the cell through the channel, which further increases the membrane potential. The ball and chain then automatically closes shortly after the sodium channel was activated, preventing more sodium from entering through that particular channel. Since the membrane potential is even higher now, more voltage-gated sodium channels open up, driving the membrane potential up even higher, even reaching a positive membrane potential. However, eventually all of the sodium channels are plugged up by the ball and chain, making them inactive and unable to let more sodium through. This causes the action potential to reach its peak. So this upswing of the action potential is driven by sodium ions entering the neuron, and this peak is driven by the inactivation of every voltage-gated sodium channel. Around this time, the voltage-gated potassium channels open to let potassium out of the cell. These channels were actually activated at the same time as the voltage-gated sodium channels, but have a small delay before finally opening. With positive charges leaving the cell, the membrane potential becomes more negative again. At some point, the membrane potential is negative enough for the sodium ion channels to close, which pushes the ball and chain out and allows for further activation. Meanwhile, the potassium channel continues to let potassium ions out, which causes the membrane potential to actually dip below the resting membrane potential. This means that the downswing of the action potential is driven by potassium ions leaving the neuron. Once all the potassium channels close, the membrane potential will return to rest. Part of this return to resting membrane potential is driven by the sodium-potassium pump. I didn't draw that here, but remember that it is always working in the background. While the action potential has finished in this section of the axon, the sodium ions that came through earlier can travel a bit further down the axon and activate another cluster of voltage-gated channels, causing another action potential further down the axon. And so on. This is how a signal gets transmitted down the axon into the axon terminal. Here we have a picture of the axon terminal and a skeletal muscle cell. The area between the neuron and the skeletal muscle cell is called the synapse. A synapse is generally any connection between a neuron and another cell. Since this is between a neuron and a skeletal muscle cell, this is specifically referred to as the neuromuscular junction. There are also voltage-gated calcium channels in the axon terminal, as well as vesicles filled with neurotransmitters, in this case, acetylcholine. As mentioned, the sodium travels down the axon and into the axon terminal. This opens up the voltage-gated calcium channels and allows calcium to enter the axon terminal. There, it contributes to the vesicles fusing to the membrane so that they can release acetylcholine into the neuromuscular junction. Before we talk about what acetylcholine does, let's take a closer look at the skeletal muscle cell. We see that it has a sarcoplasm, which is just a cytoplasm in a skeletal muscle cell, and also the sarcoplasmic reticulum, which is the endoplasmic reticulum in a skeletal muscle cell. We also see the acetylcholine receptor, which in skeletal muscle cells is a ligand-gated ion channel. This means that when a ligand binds, in this case acetylcholine, the channel will open up and let ions through. Similar to neurons, skeletal muscles also have voltage-gated sodium channels. These transmit signals down a structure called the T-tubule. In the T-tubule, there's a specialized receptor called the DHPR, which is a voltage-gated channel that opens up when it senses an increase in the membrane potential. While it is technically a calcium channel, it doesn't actually let any calcium into the cell. Instead, it is physically connected to the ryanodine receptor, which is a calcium channel on the sarcoplasmic reticulum. So when DHPR opens, the ryanodine receptor also opens. As always, there are a lot of sodium ions outside the cell. There is also calcium outside the cell, but the skeletal muscle cell doesn't really use that calcium. Instead, it uses the calcium ions that are stored inside the sarcoplasmic reticulum. There are also voltage-gated potassium channels and potassium ions inside the cell, but I didn't draw them to save space. Now let's talk about excitation-contraction coupling in the skeletal muscle cell, which is how a signal that excites the muscle cell leads to contraction. The first step is that once acetylcholine binds to its receptor, this triggers the channel to open. Sodium is the main ion that flows through this channel, which causes the positive charges in the muscle cell to increase, making the membrane potential less negative. Just like in the neuron, this change in the membrane potential triggers voltage-gated sodium channels to open, allowing more sodium into the muscle cell. Then, just as voltage-gated sodium channels open along the axon and allow sodium ions to flow down the axon, the voltage-gated sodium channels in the muscle cell open up and allow the sodium ions to flow down the T-tubule. Once the flow of the sodium ions reaches the DHPR, it will cause the DHPR to open up, which in turn causes the ryanodine receptor to open for a brief moment. This is enough time to allow several calcium ions from the sarcoplasmic reticulum to leave and go into the sarcoplasm. Now let's zoom into what's happening in the sarcoplasm once calcium is released. Here I have a portion of a sarcomere for simplicity. These blue circles represent actin, which forms the thin filament. These are the myosin, which form the thick filament. These lines covering the actin are tropomyosin, which is important for preventing myosin from binding to actin when it's not needed. These yellow triplets are troponin, which is associated with the tropomyosin. When the concentration of calcium in the sarcoplasm increases, the calcium will bind to the troponin. This causes the troponin to change its conformation, and that will actually move tropomyosin out of the way. This allows myosin to bind to actin, and as soon as that happens, contraction will occur. Zooming out a bit, let's review how these steps lead to contraction. Calcium binds to troponin, and this moves tropomyosin out of the way of myosin. Now myosin can bind to the actin. This immediately leads to contraction of the sarcomere. Note that when the sarcomere contracts, while the length of the thin and thick filaments stay the same, the amount of space between the thin filament decreases, and so does the length of the entire sarcomere. When the concentration of calcium decreases in the sarcoplasm, the tropomyosin will slide back in place when the myosin lets go, and this will let the muscle relax. That's it for this video. Thanks for watching.